You can bag or cover the stigmatic surface with a polythene paper or a polythene bag to avoid the falling of the unwanted pollen grains. So that method is called, as I said, it is also called bagging. Flower is a fascinating organ of angiosperms, and it is a modified shoot of a plant body which is meant for reproduction. The pollen grains are preserved in liquid nitrogen at -196 degrees centigrade, which are used as pollen banks in crop breeding programs. Hello everyone, welcome back to the revision class of second PUC of biology, that is chapter 2 of your biology syllabus called Sexual Reproduction in Flowering Plants. Myself, B. Shobha Rani, faculty in biology from Vidhyaashram Pre-University College, the Temple of Excellence. So what am I going to discuss in this particular session of chapter 2 called Sexual Reproduction in Flowering Plants? In this particular chapter, I'll be discussing about the important concepts of this particular sexual reproduction in flowering plants. What is the weightage of marks for this particular chapter? As I said, the first chapter called Reproduction in Organism, the basics what you studied is very important, which is going to carry forward for the remaining 3 chapters of the same unit called Reproduction. So one such chapter is Sexual Reproduction in Flowering Plants. The weightage of marks is 8 marks. So it is a big number. And what will be the type of questions that are going to be asked in this particular chapter? Like you have 3 marker question 1 and 5 marker question 1. As I keep telling you all that usually in the essay type questions, like may it be for 3 marker or 5 marker question, you may have split questions. For example, for 3 marker you may have 2 plus 1, or for 5 marker question you may have 2 plus 3. So this is how you are going to get even for this 3 marker question and 5 marks, or maybe for full 3 marks, or maybe for full 5 marks. Like for example, explain with a neat label diagram the TS of young anther. So it might be for 5 marks. Or explain the structure of a pollen grain. It might be for 3 marks. Or it might be for 2 plus 1, or it might be for 2 plus 3 for 5 marker questions. So this is how the questions are going to appear in this particular chapter called Sexual Reproduction in Flowering Plants. So weightage of marks is 8 marks. So let me move on to the introduction of this particular chapter called Sexual Reproduction in Flowering Plants. So all of us know flower. Flower is a fascinating organ of angiosperms, and it is a modified shoot of a plant body which is meant for reproduction. And when we study the structure of a flower, when we study the longitudinal section of a flower, we come across 4 very important parts of a flower, that is calyx, corolla, androecium and gynoecium. Calyx we write it as K, corolla we write it as C, androecium A and gynoecium G. So when we study the structure of a flower, 4 very important parts of a flower are calyx, corolla, androecium and gynoecium, where calyx and corolla are called non-essential orals. They are called non-essential orals, whereas androecium and gynoecium are called essential orals. Essential orals of a flower. Why are calyx and corolla called non-essential? Why these 2 are called essential orals? Because androecium and gynoecium are directly taking part in the process of reproduction, hence you call them as essential orals. Whereas calyx and corolla are helping the process of reproduction indirectly, hence they are also called as non-essential orals, or also called as accessory orals. Accessory orals. So what are the 4 orals of a flower? 4 orals means 4 very important parts of a flower. Calyx, corolla, that is non-essential orals or accessory orals. Androecium and gynoecium, that is essential orals, which directly take part in the process of reproduction. So we all know that flower is a very important, fascinating organ of an angiosperm which takes part in the process of reproduction and helps in the continuity of the plant species from generation after generation. Now, in this particular chapter, we are going to study about the structure of an anther as seen in a cross section. So, when we study about the structure of an young anther, TS of an anther, there are 4 very important walls, that is epidermis, then endothecium, middle layer, and tapetum. The first three, that is the epidermis, the endothecium, and middle layer, perform the function of protection and also help in dehiscing the anther to release the pollen grains, whereas tapetum helps in nourishing the pollen grains. Then we come across tapetum. As I said, it is very important for nourishing the pollen grains. So next we will be studying about the process of microsporogenesis, section and formation of pollen grain. The process of formation of pollen grain from the pollen mother cell, you call it as microsporogenesis. So when we study about the structure of a pollen grain, as we know, pollen grain is a male gametophyte which measures around 20 to 25 micron in width, and the pollen grain is made up of two layers, outer exine and inner entine. Exine is a discontinuous layer having a very important structure called sporopollenin, which is resistant to high acids and alkali, and we have the inner entine, which is a continuous layer, and on the exine layer there are small openings called germ pores where we don't come across the sporopollenin, and the germ pores help in to dehisce, to open and release the pollen grains. So all that you have studied here, and describe the structure of ovule, that is megasporogenesis, and formation of a female gametophyte, that is the ovule. So in this particular, you will be studying about the structure of a TS of an anther, process of microsporogenesis, structure of pollen grain, structure of ovule, that is megasporogenesis, microsporogenesis, megasporogenesis. Then explain the different types of pollination. So as we know, the very important is gamete transfer. As we all know that plants are stationary, so there must be some mechanism to help the gametes to come close together and fuse and help in the process of fertilization, and that very important event we call it as pollination. So what is pollination? You will be studying. Pollination is nothing but the transfer of pollen grains from the anther of a flower to the stigma of the same flower, when you call it as self-pollination, or different flower we call it as cross-pollination. And there are agents who bring about the process of pollination, may it be insect, may it be bird, or may it be wind, water, etc. So you'll be studying about the agent, the different agents of pollination, and the adaptation of the flowers have for a particular type of pollination. For example, like pollination through wind. So how should a flower be when it has to be pollinated through wind? It should be non-sticky, lightweight. So all that features you are going to study in this particular chapter called sexual reproduction in flowering plants. Then explain double fertilization. So as we know, the ovule contains 7 cell stage, 8 nucleate cell stage, where 3 are called antipodal cells, which are towards the chalazal region, and 3 towards the micropylar region, where 2 are synergids and 1 is egg cell, and the middle polar nuclei. So all that you will be studying here, explain, that is about the double fertilization. So then explain the development of embryo from zygote along with the diet. That is what we call it as embryogenesis. Embryogenesis. Then describe the types of fruits. As we know, there are simple fruits, then we have this multiple fruits, and fruit is nothing but a ripened ovary, right? So all that you'll be studying in this particular chapter. Then explain apomixis, that is development of seed without fertilization, and the seed that is developed is called apomictic seed, polyembryony with suitable examples. Then you'll be studying about pre-fertilization events, that is the structure and events, where you'll study about the structure of a stem and already I explained microsporangium, pollen grains, pistil, megasporangium, embryo sac, pollination, as I said, kinds of pollination, agents of pollination, that is pollination through insects, we call it as entomophily, pollination through wind, you call it as anemophily, pollination through birds, we call it as ornithophily, then pollination through animals, we call it as zoophily, and pollination through ants is called myrmecophily, then pollination through snails is called malcophily. So like this we have different agents which bring about the process of pollination. Then outbreeding devices. See how the plant will avoid self-pollination and enhance cross-pollination. So plants that are produced through cross-pollination will have a better genetic variety than the plants that are produced through self-pollination. So it will come out with some outbreeding devices that the stigma and the release of pollen grain will be at two different times, so that it can avoid, particularly when we come across a bisexual flower. So in that case it will avoid the unwanted pollen grains to come and fall on the stigmatic surface. Then once the pollen grain fall on the stigmatic surface, and when the stigmatic surface is receptive, so then what are the events that is going to happen after the pollen grains land on the stigmatic surface? You call it as pollen pistil interaction. Then we come across artificial hybridization, where it is obtained through emasculation and bagging. Emasculation and bagging. So with these two techniques, emasculation means what? Removal of stamens, or removal of stamens. Then bagging, covering the stigmatic surface with a polythene bag. So so suppose if it is a bisexual flower, so you want to allow the pollen grains of your desire to fall on the stigmatic surface and allow the process of fertilization to happen. So in that case, you can remove the stamens, and also if you want to avoid the unwanted pollen grains to fall on the stigmatic surface, you can cover the stigmatic surface with a polythene bag. And so all these are called artificial hybridization. So all that you are going to study in this particular chapter. Then post-fertilization structure, that is the process of formation of endosperm, where based on that we call it as endospermic seeds and non-endospermic seeds, right? Then embryo, then seed. We'll be studying about seed. As I already said, apomixis and polyembryony, development of seed without fertilization and polyembryony. So all these things you are going to study in this particular chapter called sexual reproduction in flowering plants. So let us discuss what are the type of questions that are going to be asked in this particular chapter. As I said, though it is 3-marker question 1 and 5-marker question, as the weightage of marks for this particular chapter is 8 marks. But as I said, for 3-marker questions, you may have split questions like 2 plus 1. So let us see what might be the 2-marker questions that are going to be asked. How are pollen grains preserved? Answer is, the pollen grains are preserved in liquid nitrogen at minus 196 degrees centigrade, which are used as pollen banks in crop breeding programs. So, how are pollen grains preserved? The pollen grains are preserved in liquid nitrogen at minus 196 degrees centigrade, which are used as pollen banks in crop breeding programs. Next, papaya plants exhibit xenogamy only. Why? So, as we know, papaya plant is a dioecious where male and female flowers are developed on different plants. So, obviously it will be xenogamy. Then, name the protective substance present on the pollen envelope to tide over adverse conditions. So, as I said, the outer exine of the pollen grain has sporopollenin, which is highly resistant to high acid and alkali. So, it can withstand high acids and alkali, and that is none other than sporopollenin. So, name the protective substance present on the pollen envelope to tide over adverse condition, that is sporopollenin. Then, explain for 3-marker questions. So, explain any 3 inbreeding devices in flowering plants. So, following are the inbreeding devices. So, there is synchrony between pollen release and stigma receptivity, that is the anthers and pistils mature simultaneously. The anthers and stigma lie close to each other in such a way that pollen from the anther can fall on the stigma. Clistogamous flowers, as we know, the bisexual flowers which do not open at all are produced by some plants. So, all these are the inbreeding devices in flowering plants, which might be asked for 3-marker question. Write the mode of pollination in Vallisneria and water lily. Explain the mechanism of pollination in Vallisneria. So, we all know that Vallisneria is a hydrophytic plant, or it is a plant which is pollinated through water, hydrophily. So, Vallisneria is pollinated by water and water lily is pollinated by insects. So, here they have asked, write the mode of pollination in Vallisneria and Water lily. Vallisneria is pollinated by water and water lily is pollinated by insects. So what is the mechanism of pollination in Vallisneria? In Vallisneria, the male flower, which we later call it as pollen grains, are released on the surface of the water. It has a long stalk where the pollen grains are released on the surface of the water and the water current carries the pollen grains. Whereas the female flowers reach the surface of the water. As you know, they have a long stalk. So what they do, by uncoiling of the long stalk, the female flowers reach the surface of the water. So along with the water current, the male, the gametes, that is the pollen grains, flow and they come and land on the stigmatic surface. So when the passively floating male flowers meet the female flowers, pollination is effected and stalk of the female flower coils and the flower is drawn back inside the water column. So this is the mechanism. This is very important from examination point of view. This is which is usually asked in your board examination. So what is the mechanism of pollination in Vallisneria? So moving on to the 3-marker question, explain 3 outbreeding devices. So as I said, the plants avoid self-pollination and enhance cross-pollination. So what are the devices the plants will try to implement to avoid self-pollination and to enhance cross-pollination? That is what is that. And this is a very important question from examination point of view. This is usually asked for the 3-marker question. So you need to explain any 3 outbreeding device. So for each device that you write, you'll be getting 1 marker. So it's like almost like a split question. So explain 3 outbreeding devices. The outbreeding devices are as follows. Self-incompatibility is the genetic mechanism that prevents self-pollen from fertilizing the ovule by inhibiting pollen germination or retarding the growth of pollen tube. So, you know, the stigma receptivity, it is called self-incompatibility. The stigma receptivity, even if the pollen grains come and fall on the stigmatic surface, it will not receive or it may avoid the process of germination. So, this is how. So, self-incompatibility is one such mechanism which is designed by the plant to avoid self-pollination and to enhance cross-pollination. Then second device is the release of pollen and the stigma receptivity are not synchronized, will not happen at the same time. So maybe when the stigma is not receptive at that point of time, the pollen grains may come and land on the stigmatic surface. So in that case, the pollen grain will not germinate. So pollen release and stigma receptivity are not synchronized. Either the anther may mature first or on the pistil or gynoecium may mature first. So the maturity of the reproductive organs also may take place at different times so that it can avoid self-pollination and enhance cross-pollination. Next, the anther and stigma of the flower are placed in such a way that the pollen of the flower cannot fall on the stigma of the same flower. So this is one mechanism wherein the size, the length of the pollen, that is the stamen, will not match with the stigmatic surface. So the height will not match, so much so that the anthers and the stigma of our flowers are placed in such a way that the pollen of the flower cannot fall on the stigma of the same flower. So these are the 3 outbreeding devices which a plant will try to implement to avoid self-pollination and to enhance cross-pollination. Then for 5-marker question, draw a diagram of a sectional view of a monocot seed. So this is the diagram where you can draw. So where you have to mention the parts also. As I said, in PUC, very important is you need to draw a labeled diagram. Unlabeled diagram or unlabeled illustrations do not carry marks. So you can see the endosperm, then scutellum, coleoptile, plumule, radicle and coleorhiza. So here it is a 5-marker question. So this is asked for 2 marks. Diagram. Draw a diagram of a sectional view of a monocot seed. Write the function of coleoptile, coleorhiza, endosperm, right? For this you will get 3 marks. So it is almost like a split question, A and B. So A 2 marks and B is 3 marks. So as I said, for 5 marks, you may get for 2 plus 3. That is for 5. So diagram 2 marks. Then write the function of coleoptile. So what is the function of coleoptile? Protects the plumule. Then coleorhiza protects the radicle. Endosperm stores food materials to be used by the embryo during germination of the grains. So this is how the 5-marker questions are asked in your board examination. Then explain double fertilization. As I said, the pollen tube, as I said, once the pollen grain come and land on the stigmatic surface, there will be a very important event called pollen pistil interaction, where we come across the pollen tube discharges after germination, the pollen tube carries two male gametes, and after entering one of the synergies. So one of the male gamete enters the embryo sac through the synergids, and one of the male gamete fuses with the female gamete to form a zygote, and this is called syngamy. So one of the male gamete fuses with the egg cell to form a zygote, and the other male gamete fuses with the secondary nucleus to form the primary endosperm nucleus. So this whole process, you call it as double fertilization and triple fusion. As we know, the secondary nucleus will be 2n and one of the male gamete is n, so it will be 3n. So hence you call it as what? Triple fusion. So you need to explain the process of double fertilization. Then 5 marker questions like, what is artificial hybridization? Explain the process of artificial hybridization. So what already I have discussed in the previous question, artificial hybridization involves crossing of different varieties or species and even different genera to combine the desirable characters of the two parents to produce a commercially superior variety is called artificial hybridization. So what is artificial hybridization? Artificial hybridization involves crossing of different varieties or species or even different genera to combine the desirable characters of the two parents to produce a commercially superior variety. Very important, right? How is it achieved? Artificial hybridization is achieved by two very important techniques, methods. One is called emasculation, the other one is called bagging, which I am going to discuss in the coming slide. So emasculation, the very word itself will tell you. And this emasculation is needed when the female parent bears the bisexual. So if it is a bisexual flower where we come across both the male and the female flower, so the chances of pollen grains falling on the stigmatic surface of the same flower is more possible. So as I said, you want to dust the pollen grains of your desire on the stigmatic surface. So you need to avoid the falling of unwanted pollen grains on the stigmatic surface. So for that case, what you have to do is removal of anthers, stamens, before the pollen grains mature. So that process is called as what? Emasculation, removal of stamens. In simple terms, if you want to explain what is emasculation, it is nothing but removal of stamens. Removal of stamens is called emasculation. So the emasculated flower is bagged to prevent contamination of the stigma with unwanted pollen grains. So if it is a bisexual flower, though you have removed the stamen, but there are chances of some XYZ pollen grain coming and falling on the stigmatic surface. So in order to avoid that also, what we can do is, you can bag or cover the stigmatic surface with a polythene paper or a polythene bag to avoid the falling of the unwanted pollen grain. So that method is called, as I said, it is also called bagging. So even if it is emasculated, we need to cover the stigmatic surface with a polythene bag to avoid the falling of unwanted pollen grains. So when the stigma retains receptivity, so as we know, only when the stigma is receptive, it is capable of accepting the pollen grain of its desire, if its own species, then only the pollen germination can take place. So once when we get to know that the stigma is receptive, the pollen grains collected from the selected male parent are dusted on the stigma. So this is how you can produce a plant of your desire, and that is what we call it as artificial hybridization. So next is, the flower is rebagged and allowed for further development. So when the female parent bears only female flower, then again I said, when it is a unisexual female flower, the chances of XYZ pollen grain coming and falling on the stigmatic surface is quite possible. So you need to bag it. So bagging is needed. So no emasculation, just bagging, so that you can avoid the falling of the unwanted pollen grains. So the female flower is bagged before the stigma becomes receptive. So when the stigma becomes receptive, as I said, the pollen grains collected from the selected male parent are dusted on the stigma, and the flower is rebagged, again rebagged, and allowed for further development. So these two very important techniques are applied in artificial hybridization to produce a superior variety of a plant. So let us recap the whole chapter, that is sexual reproduction in flowering plants. So as I said already, so this flow chart will give you what are all the things you are going to study in this particular chapter called chapter 2 of your second PUC biology syllabus called sexual reproduction in flowering plants. So as we already discussed that flower contains 4 very important parts, that is calyx, corolla and androecium and gynoecium. So as we know, as I already discussed, flower has calyx, corolla, androecium and gynoecium. Androecium is called the male reproductive part of a flower and gynoecium is called as a female reproductive part of flower. So what are the parts of a gynoecium? You come across stigma, style, ovary and ovules. What are the parts of an androecium? Stamen, filaments and anthers. Right. Now, when we come across, as I said, 2 very important processes are happening, that is microsporogenesis and megasporogenesis, right? Microsporogenesis and megasporogenesis. So here you can see the formation of, as I said, in double fertilization, you come across 7-celled, 8 nucleate stage, where 3 of the cells are towards the chalazal region called antipodal cells and 3 towards the micropolar region, where 2 are synergids and 1 is X cell, and in the center you come across the 2 polar nuclei. So all that we have, as I said, the fusion of, as we call it as double fertilization and triple fusion. Double fertilization and triple fusion. So 8 nucleated, 7-cell stage. So the 3 antipodal cells will disintegrate, whereas the 2 synergids will allow the 2 male gametes that are carried by the pollen tube to enter the embryo sac and fuse, one of the male gamete fuse with the X cell to form a zygote. So synergids later will also dissolve, and the 2 polar nuclei, which are 2n in number, will fuse with one of the male gamete to produce the primary endosperm nucleus, so which is 3n. Hence I said it is called double fertilization and triple fusion. So as I said, pollen grain, which is a very important structure, male gametophyte, which has outer exine and inner entine. Outer exine is discontinuous, having germ pores where you don't come across the sporopollenin. Sporopollenin is found on the exine layer, which is very important, which is highly resistant to high acids and alkali, and which can withstand any adverse climatic conditions. So the inner layer intine is continuous layer, right? So all that we have discussed. And what was pollination? As I already said, pollination is very important for the gametes to come together. As we know, plants are stationary. So pollination enables the pollen grain to come and fall on the stigmatic surface of the female flower and allow the process of fertilization to happen. So all that we have discussed in this particular chapter called sexual reproduction in flowering plants. Hope you all have understood the small revision part of this particular chapter called sexual reproduction in flowering plants. This chapter is very important from examination point of view, as it carries 8 marks, which is a big mark. So hope you have understood what might be the type of questions, which are briefed up as to what type of questions will be asked. Like if you go deeper into the concepts of this chapter, then you will get to know more number of questions, like how they are going to appear in the board examination. So thank you.